replication of DNA. In this section we're going to get into talking about DNA, RNA, and proteins. So we'll start out the lesson with a bell ringer. We'll look at the key ideas. We'll talk about DNA replication, replication proteins, prokaryotic and eukaryotic replication, and then we will summarize the entire section. So your bell ringer. DNA is considered to be a relatively stable molecule, meaning that it doesn't break down very easily. What gives it this stability, even though the hydrogen bonds between the nitrogen bases are easily broken? Remember in the past we've talked about how DNA goes through replication. Every time your cells divide, there has to be an exact copy of the DNA that is in it. So how is it that those bonds are easily broken so that then replication can occur, but DNA is relatively stable even with those weak bonds? Write out your answer in your warm-up sheet and then share and compare your ideas with other students as well. Key ideas. How does DNA replicate or make a copy of itself? What are the roles of proteins in DNA replication? And then how is DNA replication different in prokaryotes and eukaryotes? Remember, a prokaryote is an example of an organism that does not have a membrane-bound nucleus. Eukaryotes are organisms that have a number of membrane-bound organelles. Prokaryotic examples would be things like bacteria. And bacteria really only have ribosomes floating around on the inside, and then all the DNA is kind of just mixed into the cytoplasm inside of a bacterial cell. Whereas in eukaryotes, all of the DNA is housed inside of the nucleus. And then remember that there are nuclear pores where mRNA can help by making copies of that DNA, and the mRNA transfers that information out to the ribosomes, where the proteins are assembled. DNA replication. Because DNA is made of two strands of complementary base pairs, if the strands are separated, then each strand can serve as a pattern to make a new copy. So it's kind of like a zipper. If we can take and we can make copies of each strand, then we're going to end up with a complementary strand that is exactly the same as the previous strand. The process of making a copy of DNA is called DNA replication. And remember, this process occurs during mitosis inside of the cells. In DNA replication, the DNA molecule unwinds and the two sides split. Then new bases are added to each side unit, forming two identical sequences. So a whole bunch of extra bases are kind of assembled right on top of that unzipped section of DNA. So as the double helix of DNA unwinds, the two complementary strands of DNA separate from each other and they form Y shapes. Those Y shapes are called replication forks. So it's where the DNA unzips. As it is unzipping, that area where the two halves are pulling apart, they are the replication fork. That location is called the replication fork. At the replication fork, new nucleotides are added to that DNA, added to those complementary strands. And those new nucleotides that are added, they're added according to the base pairing rules. So remember back to when we talked about what nucleotides bind with what nucleotides. The nucleotides, remember, are adenine, guanine, cytosine, and thymine in DNA. So each double-stranded DNA helix is made of one new strand of DNA and one original strand of DNA. And the next slide is going to show visually how this process occurs. Alright, so first step in making a copy of DNA includes helicases. Helicase is a protein that attaches to the DNA. That protein helps to break the hydrogen bonds between the nucleotide sequences. So DNA helicase attaches and it basically breaks those bonds and unzips the DNA. Complementary nucleotides are then added to each strand of DNA polymerases. So DNA polymerase jumps on. DNA polymerase is used to attach new nucleotides, so we end up with two complementary strands. So the DNA polymerase basically follows the DNA helicase as it unzips, and it just adds new sections on as that unzipping occurs. And then two DNA molecules are formed that are identical to the original DNA molecule when this entire process is over. So the replication of DNA involves a number of proteins. We just saw those proteins in the last image. DNA helicases, they unwind the double helix of DNA and they create that replication fork. They basically wedge themselves between the two strands and then they break those hydrogen bonds. And then DNA polymerase, they help to catalyze the formation of the DNA. They basically move along behind the helicases and they add new nucleotide pairs to the existing strand of DNA. So it ends up creating two completely new strands of DNA. DNA polymerases also have kind of a proofreading function. They go through and they match up the base pairs dependent upon what was in the existing strand of DNA. And during replication, errors sometimes occur where the wrong nucleotides are added to the new strains. And if that mismatch occurs, the DNA polymerase can backtrack and it can remove the incorrect nucleotide that was added. So it ends up correcting itself in this process of making copies of DNA. So this is one way in which your body keeps from making mistakes, where your body doesn't accidentally produce a mutation that causes an abnormal protein to be built or a different hair color or whatever it may be.
DNA replication begins when enzymes called helicases unwind the DNA into two separate strands. Next, enzymes called DNA polymerases move along the separated chains, assembling new chains using complementary nucleotides. The result of replication is two identical DNA molecules, each made up of an original strand and a new strand.
Prokaryotic and eukaryotic replication. So all cells have chromosomes, but eukaryotes and prokaryotes, they replicate their chromosomes very differently. In a prokaryotic cell, remember prokaryote would be an example of like a bacteria, replication starts at a single site. So there's only one place where that DNA helicase comes on and attaches to the DNA. In eukaryotic cells, replication will start in many places along the chromosome. So it ends up being a lot faster because a whole bunch of different proteins, a whole bunch of different helicases attach to the chromosome, to the DNA in different locations, and it can speed up the process when many of them are working together. Prokaryotic cells usually have a single chromosome, which is a closed loop attached to the inner cell membrane. So it's basically this big long loop of DNA that just loops around. Whereas we have these big long blobs of DNA that kind of wind around each other to create the chromosomes. Prokaryotic cells usually have a single chromosome that is very easy to make those copies of. And then replication in prokaryotes begins at one place along the loop. So the site is called the site of the origin of replication, so it's the site of origin for where replication is going to occur, where those proteins are going to attach and start making copies of the DNA. So two replication forks begin at the origin of replication in prokaryotes. So basically it's unzipping in two opposite directions. Replication occurs in opposite directions until the fork meets on the opposite side of the loop. So basically that protein just kind of winds its way all the way around the loop until it comes back to its point of origin. In eukaryotic cells, there's several chromosomes which are linear, and they contain both DNA and protein, and the replication starts at many sites along the chromosome. So the process allows our cells to replicate the DNA much faster than prokaryotes. So our cells are very efficient at copying the DNA. So for us, we get two distinct replication forks form at each site where replication starts. So anytime that protein attaches to the DNA, wherever the location is that a protein attaches to the DNA, that would be the start site, and we get two replication forks at that site. The process forms replication bubbles. So there's these small areas where just kind of this bubble forms where the DNA is getting copied. And replication bubbles continue to get larger and larger as more and more of that DNA is copied. As more sections of that DNA slowly are copied, they just kind of slowly pull apart from one another. So here's an example of prokaryotic and eukaryotic replication. This picture shows prokaryotic DNA. So right here we got two replication forks where the helicase is basically just unwinding in one direction. It might start over here and work this way, and eventually it'll come back to its original point of origin for the prokaryotic DNA. And then in eukaryotic DNA, we'll have a whole bunch of locations where those helicases would attach. So it's attaching here, and it would just unzip, and then behind it, the polymerase would come by, and it would attach more nucleotides, and it would slowly unzip this strand of DNA. But it happens not in just one spot, it happens in numerous spots, creating these replication bubbles. So we end up with new DNA being formed on the inside of each one of the replication bubbles here. I think we've gone through everything else that's on this slide. We got new DNA, the kind of a light green color right there. The original DNA is the DNA that was the blue color. And then over here our original DNA is also blue in the eukaryotic cell DNA. So this image helps to explain why DNA replication in eukaryotes can be much faster than prokaryotic DNA replication. When you've got numerous proteins that are attaching and creating new strands of DNA, the workload basically gets diminished, so it happens much quicker. The smallest eukaryotic chromosomes are often 10 times the size of prokaryotic chromosomes. So our chromosomes are much larger than any prokaryotic type of chromosome. Eukaryotic chromosomes are so long that it would take 33 days to replicate a typical human chromosome if there were only one site of origin. That would be a long period of time for your DNA to make copies, which would slow down cell division, so you'd grow much, much slower. And then outside of that as well, if you were to get injured and need to make copies of cells for repair, that process would happen much slower than what it does on our bodies as well. Human chromosomes are replicated at about 100 sections that are 100,000 nucleotides long, each section with its own starting point. And because eukaryotic cells have multiple replication forks working at the same time, an entire human chromosome can be replicated in about 8 hours. So from 8 hours to 33 hours, or 33 days, that's a pretty drastic difference in the amount of time that replication can occur. So in summary, in DNA replication, the DNA molecule unwinds and the two sides, they split apart. Then new bases are added to each side until two identical sequences result. The replication of DNA involves many proteins that form a machine-like complex of moving parts. And those proteins include DNA polymerase and DNA helicase. Remember, helicase unzips. Polymerase then comes in and adds new nucleotide sequences, new base sequences to create new strands. In prokaryotic cells, replication starts at a single site. In eukaryotic cells, replication starts at many sites along the chromosome. So eukaryotic cell replication is much faster than prokaryotic cell replication.